as you inject more of the drug, it starts affecting motor function. So you talk like this when you get a large dose. And in many instances, it takes a number of hours for the drug to wear off. So physicians give different types of anesthetics, like they can give spinal blocks where they inject those drugs that affect the spinal nerves, and they can literally give regional blocks to different parts of the body. All right, so this shows the example of how myelin is found in nerves, and you typically have zones or regions that lack the myelin. Those are called nodes of Ranvier. And there's what is known as saltatory conduction, so the myelin prevents the leakage of charge, so it allows the signal to progress relatively rapidly. So some of the fast pain signals that are transmitted via the sensory nervous system travel via these myelinated nerves, whereas slow pain, for instance, if you stub your toe, initially you feel the sharp pain, but then you start feeling the throbbing. The throbbing is an example of slow pain. And though that slow pain is transmitted by unmyelinated fibers, and it travels much slower. All right, so the electrical synapses, we talked before about gap junctions. We'll see in the heart, gap junctions allow the wave of depolarization to travel across the various chambers, like in the atria or in the ventricles. Chemical synapses, the synthesis of neurotransmitter, the calcium channels, the voltage-gated calcium channels that open when the action potential releases the end of the nerve, allows for the release of neurotransmitter. And then in the synaptic cleft, when the neurotransmitter is released, it then traverses the synaptic cleft and binds to the receptor on whatever tissue is downstream. And that binding of the neurotransmitter to the receptor then initiates the signal transduction pathway that allows the downstream cell to perform whatever function it is programmed to do based on what transduction pathway it's connected to. Okay, so this shows an example, for instance, of what a motor nerve would look like. So this is known as the neuromuscular junction. So when the action potential travels down the axon, it reaches the terminus, opens voltage-gated calcium channels. Calcium comes in, which then allows the fusion of the vesicles of neurotransmitter with the cell membrane. The neurotransmitter goes into the synaptic cleft and potentially binds to the receptor. And in the case of muscle tissue, it opens a sodium-potassium channel, allowing sodium to come into the cell and potassium to leave. And we'll see that that then initiates a sequence of events that allows the skeletal muscle to contract. And this shows it a bit more clearly. So the postsynaptic cell has the receptor. The occupancy of the receptor is going to then determine how much signal the downstream cell is going to have and what response is going to happen. The neurotransmitter can then diffuse away. It can be taken back up into the nerve that released it. It can also be degraded by enzymes that are present on the postsynaptic cell membrane. For instance, in skeletal muscle, you have an enzyme known as acetylcholinesterase. And acetylcholinesterase breaks down the acetylcholine into acetate and choline molecules. And there you can see that when it binds to the receptor, it opens sodium and potassium channels to allow sodium to come in, potassium to leave. Here is the acetylcholinesterase that breaks up the acetylcholine so it no longer is able to bind to the receptor. All right, so neurocrines, examples of neurocrines. Neurohormones are hormones that are released from neural tissue. Remember, we talked about the pituitary and the adrenal gland. So the posterior pituitary and the adrenal medulla are examples of neural tissue that release hormones. So those are neurohormones. So examples of neurohormones: norepinephrine, epinephrine, dopamine, vasopressin, and oxytocin. Neurotransmitters: serotonin, acetylcholine, all sorts of different examples of those. And neuromodulators, those are drugs that can affect the activity and potentially binding of the neurotransmitters. You can have agonists, you can have antagonists, you can have all sorts of different molecules and drugs that affect different body systems. So these are just some of the examples of those. Histamine is another example. You can see the receptor that they bind to together with whether or not they're activating a G protein coupled receptor, an ion channel, what have you, where the receptor is located, and some of the key different drugs or agents that can be used to modify the activity of those particular neurotransmitters. And glutamate is one of the main excitatory transmitters found in the central nervous system. It's actually activated monosodium glutamate, MSG, found in Chinese food. It activates the umami receptor, and it's thought that we have receptors for glutamate because we need the raw material to keep the supply of glutamate for the central nervous system. You also have all sorts of others, gamma-aminobutyric acid, different benzodiazepines, Valium, as well as Klonopin and things like that, stimulate GABA receptors, which actually are inhibitory. They open chloride channels. So they inhibit the function of many of the nerves in the CNS, and they literally help to calm you down. And I think that's a good place to stop today. So I'll finish chapter 8 on Wednesday, and then, as I said before, on Friday I'm going to give you a take-home test so that you're able to do it over the weekend. And I'll give you questions that relate to the things that we've gone over, just to make sure that you're following along and understand the topics that we've discussed. So if anybody has any questions or concerns, feel free to email me, and we'll try to make the best of this unfortunate situation. So stay safe, everybody, and we'll see you then on Wednesday.